Right. So yeah, good evening. Good evening and welcome to today's live session class on the course Experimental Biochemistry of week 8. So today we would be mostly discussing about enzyme kinetics, and then firstly I would be discussing the lecture slides in a bit briefly, and then we would be solving a few questions. Okay, so let us start with our class today. I am sharing my slide. Kindly confirm. My slides are visible to you all? I mean my screen, right? Yeah, that's good. Fine. Okay, so let us start. Okay. So till now, how much we have learned is basically about proteins, then amino acids, the purification, characterization, and the different ways of isolating proteins by natural sources and by recombinant means. How can we engineer the proteins and the different techniques we can use to separate these proteins from a mixture and carry out different functional assays. Today we are going to study a type of a protein. So on the different, there are many types of proteins, but this is a specific and a very important type of a protein which we know, which we can call them as enzymes. So what are enzymes? Enzymes are basically proteins that increase the rate of the reaction by lowering the energy of activation. So it does not interfere with the reaction. Rather, what it does, it decreases the energy of activation. That leads to the increase in the rate of the reaction. That is, it pushes the reactants to the product side. So that is very important, and that is the role of enzymes. So they are known as the biological catalysts. They are neither altered nor they are consumed during the reaction. Rather, we get back that enzyme at the end of the reaction, which can be used, cannot be reused. There are different types. Okay. So what they do, they catalyze nearly all the chemical reactions taking place inside the cells of the body. Hence, we call them as the biological catalysts. Mostly these enzymes are, or all of these enzymes are proteins that have a particular code that includes a few or more than a few amino acids, varying from 3 to a few of the helping amino acids that take part in the different catalysis process. So this is what we understand about enzymes, and they are reusable. Sometimes the enzyme gets deactivated, which we need to use some techniques to reactivate these enzymes after we use them. So they are the biological catalysts. So these are enzymes are neither consumed nor produced during the course of a reaction. They do not cause reactions to take place, but they enhance the rate of the reactions that proceed in a slower, because we are using them. Why are we using them? To increase the rate of the reaction, because probably that reaction is slow in nature. So to increase the speed, the rate of the reaction, we are using these biological catalysts. So they alter the rate, not the equilibrium constants of the reaction that they catalyze. This is a very important point that you need to understand, right? Because it takes place, but it enhances the rate of the reaction that proceed in a slower manner. They alter the rate, not the equilibrium constants of the reaction. So this is that you need to keep in mind, because there are several times where people understand this wrong, that they also change the equilibrium constant of the reaction. No, the rate is enhanced, not the equilibrium or the equilibrium constants. So what is the difference between an enzyme and the chemical catalyst is that enzymes are proteins. They are highly specific and produce only, very importantly, only the expected products from the given reactants or substrate. Okay, so that what does it mean? That there are no side reactions. Enzymes may show high specificity towards one substrate or exhibit a broad specificity using more than one substrate. So enzymes usually function within a moderate pH and a temperature range. These are the basic differences between enzymes, and so how enzymes are better than the chemical catalysts. Okay. So if from this image or from this diagram you can understand that for a normal reaction what is happening is that you can see that the energy barrier is high, and these are the four frogs. So you can see there are four frogs that cannot cross this high energy barrier. So what happens, these frogs remain on the shore, but only one frog can cross the barrier. So that indicates that the population of the frogs being on the shore and not being able to cross such a high energy barrier will be more than the number of frogs on the product side or on the lake. So this indicates that the reaction proceeds at a slow rate due to prohibitive activation energy threshold. So what does the enzyme do? It lowers the activation energy threshold, and that helps the frogs to cross this particular small lowered energy barrier, and the population on the product side or on the frogs on the lake side is more than on the dry side. So that is, it enhances significantly there is faster rate because the activation energy threshold is reduced. This is concept of free energy. So what does this free energy say? More free energy means less stable, greater work capacity. So free energy decreases direction of spontaneous change. So here more free energy indicates a positive value, so less stable, greater work capacity. But when the energy decreases, the simultaneously what happens, delta G becomes negative, that is the reaction becomes spontaneous, the reaction becomes more feasible, the direction of equilibrium is changed, and it pushes the reaction to perform more work. So from a more free energy to a less free energy means more stable, less work capacity. So what happens when the free energy of the reactant is greater than the product? The free energy changes for the reaction is negative, so the reaction proceeds spontaneously. Now what if the free energy of the product is greater than the reactant? Then the equilibrium is more towards the reactant side, and there the delta G is positive. Why is it positive? Because delta G reaction is equal to delta G of product minus delta G of reactants. We all know that, we have all studied that. So what does that indicate? That higher the energy, higher will be the value. Higher the value, higher positive will be delta G. Higher positive delta G indicates non-spontaneous reaction. So from the A side, what happens? Higher free energy on the reactant side means more negative will be delta G. More negative delta G means more feasible will be the reaction, and the delta G will definitely push the the reactants to form more and more products, which is not the case when the energy is higher for the products. So this is the entire concept of delta G. Now delta G is related to entropy, enthalpy. Rather, if I say it in a reverse manner, it sounds more scientific, where entropy and enthalpy dominates or controls your delta G, and also delta G is related to the equilibrium constant. So it's a great relation in the thermodynamic cycle. Okay. So something, the basics of what we need to know about the components of an enzyme. So you see, this is an apoenzyme where you have only the protein portion that is inactive. Now to activate this, you need some cofactors, some coenzymes that will activate this apoenzyme or only the protein portion. These cofactors can be your ions, can be activator ions, can be metal ions, can be your some prosthetic groups, can be some co-substrates, can be even huge proteins. Okay, so this is a, say, a protein which is inactive in its natural form. You need a coenzyme, say a calcium ion or a magnesium ion, say a small molecule that comes and binds to this apoenzyme. Okay, now when this coenzyme goes and binds to this apoenzyme, what happens? It activates the protein, which we call as the holoenzyme or the whole enzyme that is in its active form. And when it goes into its active form, it is able to take up its substrate. Also, structurally, if you see that this apoenzyme is not structurally uh, attracting that particular substrate. Okay, so what do I mean by not structurally attracting the substrate? It means that its structure is not properly folded or is folded in such a manner that it cannot take up or it cannot bind to another molecule. The coenzyme, what it does, it either structurally alters the folding of that particular protein, or it somewhat enhances the ability of that particular amino acids in that protein to go and bind to the substrate, thereby making that holoenzyme or the entire of that enzyme active in its active form. So, and there are other cofactors which includes your ions and the coenzymes. So you have the activator ions, the metal ions, co-substrates, and the different prosthetic groups. So that includes the entire of the components of an enzyme to how it goes and binds to a substrate, or a substrate comes and binds to a enzyme. So apoenzyme is your protein form. Your coenzyme is the non-protein portion, or rather we can call it as the activator, and then the holoenzyme is the entire of an active enzyme. So what do we mean by an active site? So it's a very interesting term that works very well for an enzyme. It is that active site. Okay, so from this name, as we can understand that the area on the enzyme where the substrate or the substrates attach is called the active site. So see, you have a binding site that holds the substrate in its place. Then you have the catalytic site where your reaction occurs, and substrate goes and binds over here. So we call that as the active site of that particular enzyme. That it is structurally mm, uh, it is structurally feasible to take up that particular substrate. So you see, you have an active site. So substrate entering the active site of an enzyme, it comes, we are considering this to be, okay, so inactive protein may or may not have an active site, but an active protein definitely has an active site, we know that. So this is the active site where your substrate comes and binds over here. So the enzyme changes shape. Just right, sorry for the yeah. So your enzyme changes the shape to accommodate that particular substrate. You get the enzyme substrate complex, and then the enzyme pushes the product formation and you get the different products. So the theory behind enzyme kinetics, you have the two kinds of models that work perfectly over here. One is the lock and key model, another is the induced fit model. So what are the concepts of these two models? We will be talking about it now. So from the name, as you can understand, lock and key. So there is a shape. You understand what happens. You have a particular lock. You put any key, it does not fit. Only that key that is made according to the lock, only that is going to fit over here. So what does that mean? That it is specific. At the same time, the enzyme cannot change its shape according to the substrate. That is a disadvantage, because because then the enzyme is only specific to that particular substrate. It is one way very advantageous. Why is it very advantageous? Because the enzyme substrate complex that will be formed is very specific to your particular substrate, because no other substrate will be able to bind over there. But if you do not know about the structure of that particular substrate or the reactant that you want to study the binding with that particular enzyme, it becomes very, very difficult. So it is a very older model that does not work for all the enzyme. So this lock and key model, in briefly, if I say, the active site has a particular rigid shape. Only substrate with the matching shape can fit into it, and the substrate is the key that fits the lock of the active site. And then you get the enzyme substrate complex towards going to the product site. However, the induced fit model is a lot more advanced model. It is consistent with a wider range of enzymes. What happens over here is that the active site is very flexible. That is, it is not very rigid, so it can change its shape according to the substrate. So the way it wants to bind with the substrate, it is going to change its shape. So the shape of the enzyme, active site, and the substrate is to maximize the fit, which improves the catalysis. And there are a greater range of substrate specificity. Not only one kind of substrate can go and bind over here. There are different kinds of substrate that goes and binds over here. Might be the specificity, from the perspective of specificity, lock and key model was better, but what the applications of induced fit model wala enzymes are far better, and they are highly advanced in terms of their ability to be flexible at the active site and encourage different kinds of substrate to bind over there and proceed towards the product site. So the applications of this kind of enzymes will be very high in different biotechnological and bioengineering studies. Right, I think this much about the theory of enzyme kinetics was clear. Now there are a few things about the classification of enzymes where you need to understand a few terms and all. So let us know about that. So what is an oxido? So the classification of enzymes can, according to the IUMB system, it's categorized into 7 types. So IUMB is International Union of Molecular Biochemists. So they are classified according to excuse me. So yes, they are classified into 7 different categories, where you have oxidoreductases, you have transferases, then you have hydrolases, followed by lyases, then isomerases, ligase, and translocases. So I would be discussing about the functions of all of these enzyme classifications. But before that, we need to understand a very important term, the turnover number. So turnover number or catalytic efficiency, what does it say? It says that most enzyme catalyzed reactions are highly efficient because it enhances the rate of the reaction, and proceeding from 10 to the power 3 to 10 to the power 8 times faster than the uncatalyzed reactions. So what happens that typically each enzyme molecule is capable of transforming 100 to 1000 substrate molecules into product per second. So the turnover number, so this is your catalytic efficiency. What does the turnover number say? The turnover number says it can be defined as the number of molecules of substrate that gets converted into the product per enzyme per second. So one enzyme in 1 second, how many molecules of the reactants is getting converted to a product. So the turnover number, if the turnover number is say 2, it means that 2 molecules of the reactants are getting converted to products by one enzyme in 1 second. I think it is clear. Now next. Okay. About the functions of the classifications of these enzymes. So we all know about oxidoreductases that catalyze oxidation and reduction reactions, or the redox reactions. Now this reaction involves the transfer of protons or electrons between the substrates. For example, we have heard of the oxidase and the alcohol dehydrogenase, and there are other examples as well. Now, transferases, from the name it suggests that it catalyzes the transfer of a functional group from one substrate to the another, and these groups can be in different categories, from starting from the methyl to the ethyl to different amino groups, phosphate groups. Then you have some ionic groups, etc. And examples includes your nucleoside monophosphate kinase, the NMP kinase. You have the transaminase. Now, hydrolyses are ones that catalyze hydrolysis. Hydrolysis means breakdown. So, breakdown reaction with water, which is the transfer of functional groups to the water, where you have the activity of the lipase, amylase, and the peptidase. In category 4, sorry. So what happens in category 4? We have the lyases. So lyases catalyze the addition of groups to the double bonds and formation of double bonds by the removal of groups. We have the fumarase, decarboxylase, etc. Then isomerases, which catalyze the transfer of groups within molecules to yield isomeric forms and carry out isomeric reactions. It catalyzes the intramolecular transfer of different molecules or groups, where we have the triose phosphate isomerase, phosphoglucomutase, the most important being ligase, which we need for the ligation reaction, the condensation, the joining of two molecules with the expense of energy from ATP hydrolysis. It catalyzes the formation of carbon-carbon, carbon-sulfur, carbon-oxygen, carbon-nitrogen, and several phosphate groups bonding by the condensation reaction coupled with uh, ATP cleavage, and the examples being amino acid tRNA synthetase, then you have the DNA ligase, and finally translocases. This class has been recently added by this particular IUMB system that catalyzes the movement of ions or molecules across the membrane or the separation within the membranes. So so one example is ATP synthase, right? So all the systems have been classified in these 7 major groups, and each major group is further subdivided into subclasses, subclasses, and a specific code has been assigned them. That code number you call as the enzyme commission number or EC number, which consists of 4 digits separated by periods. Now, for example, if I give you one example, say so the number is 1.1.1.1. So 4 ones separated by points. So the first digit indicates the major class, the second digit is the subclass, the third digit is the subclass, and the fourth digit indicates the systemic specific name of that particular enzyme. Okay, so This particular number is designated to alcohol dehydrogenase 1.1.1, 1.1.1.1. So all ones, four ones. Right. Next, we go to the environmental factors that determine the activity of the enzymes, which includes your optimum temperature, the temperature at which the enzymatic reaction occurs the fastest. That is your at 37 degrees, the human body temperature. That is why we are still very healthy. So the pH also affects the rate of the enzyme substrate complex. So most enzymes have an optimum pH of around 7. However, some prefer acidic and basic conditions. Like you have pepsine that prefers acidic condition, you have trypsin preferring basic condition, and you have the human amylase that is happy at neutral pH. Okay. Right. So the substrate concentration and the reaction rate. So the rate of the reaction increases as your substrate concentration increases. So more number of substrate, higher will be the rate of the reaction at constant enzyme concentration. So maximum activity occurs when the enzyme is saturated. So that is when all the enzymes are binding substrate. So if you see that when the concentration of the substrate is yes, when the concentration of the substrate is low but the enzyme concentrate is constant, so you see that at this point the enzyme gets saturated with all the substrate, and after this there is no enhancement in the rate, and the condition or the rate at which the saturation level is reached is known as the maximum velocity or Vm, and this is the initial reaction rate, and this is the maximum velocity where you observe the maximum activity, and this is the position where all the enzymes is combined with the substrate, means saturation is about to reach, and then here the enzyme activity increases. Now coming to the most important portion about the kinetics of enzymes. So enzymes follow zero order kinetics when substrate concentrations are high. Zero order means there is no increase in the rate of the reaction when more substrate is added. Now given the following breakdown of sucrose to glucose and fructose, you can understand. So you see you have enzyme, you have substrate, then you have the enzyme substrate complex, you have then you are getting back the enzyme, and then you get the product. In between you have this an equilibrium position where you have K1 constant for the forward reaction, you have K minus 1 for the rate constant for the breakdown of the enzyme substrate to substrate, and finally K2 the rate constant for the formation of the products. Now when the substrate concentration becomes large, there is enough to force the equilibrium to form completely all the enzyme substrate complex. So once the enzyme substrate complex is formed, there is no pressure of the products been forming, because they will definitely form the products. So the step, that is the rate limiting step. So what is the rate limiting step? That controls What does it control? It controls the formation of products and the rate of the reaction, whether the reactant is forming to the product. That's an intermediate step. And this intermediate step, what happens in this intermediate step is that you have the enzyme substrate complex. You may or may not be able to isolate that particular complex, but what matters is that the concentration of this enzyme substrate according to the steady state approximation is considered to be zero, because we are considering that the substrate is entirely getting converted to the product. In the middle you have a complex that is utilized to get to the product. So the rate of this intermediate product is considered to be zero. So this is the rate where you have dp by dt equals to k2 into substrate concentration. That is the difference between the rate of the enzyme substrate formation minus the rate of its disappearance. So if you see that d of es enzyme substrate complex by dt, that is the rate, equals to k1 into enzyme into substrate concentration minus k minus 1 into this complex concentration and minus k2 into this complex concentration. How are you getting this? So looking from the perspective of this enzyme substrate complex, what do you see? That when the enzyme and substrate is getting consumed to get to the complex, so this is the forward reaction. So forward reaction means the complex is getting formed. So the complex is getting formed, that is why it is positive. Where the complex is getting utilized, where it is getting utilized, it is getting utilized in the second step and the first. That is the backward reaction. The backward reaction and in the second step. So in both these steps you see that the sign is negative. But in the other two steps, in the other step it was positive, because the complex was getting formed. The complex getting formed positive, the complex getting utilized negative. And from here you can get, so according to the steady state approximation, the des by dt equals to 0. And if you assume that k minus 1 greater than k2, so the formation of product is so much slower than the formation of the complex, that we can assume that Ks, that is the dissociation constant of the enzyme substrate complex, the dissociation constant for the enzyme substrate complex is equal to K minus 1 by K1, that is equal to enzyme concentration minus into substrate concentration divided by the enzyme substrate complex. So another assumption is the steady state approximation. So transient phase, where in the course of the reaction the concentration of ES does not change, so we consider that dES by dt is close to 0. This is very important to know, because in any step, the intermediate step where it is in the middle and from substrate you are getting to the product, and also we need to consider it as the rate limiting step that controls the rate of the reaction. The steady state approximation stands where dES by dt is equal to 0. You can see the calculation over here, where the total enzyme concentration is equal to the enzyme concentration plus the enzyme in the substrate concentration. And then when you combine, you have several equations over here. When you combine all of these equations and rearrange it, you get some equation like this, where you have the complex followed by a few constants into the substrate concentration. Then you have K1 into the total enzyme concentration and the substrate concentration, and finally you come to this. So now when you substitute all of this, or you do not need to substitute your K1 Km, because you need to substitute this portion. So Km equals to minus K minus 1, that is the backward rate constant, plus K2 divided by K1. finally, this is the initial rate where dp by dt is equal to 0, and this is the product formation. This is the rate of product formation, where v naught is the initial velocity when the reaction is just starting out. So finally you have Vmax equals to K2 by total enzyme concentration, and this is the maximum velocity. So you see maximum velocity by 2 equals to maximum velocity into substrate concentration divided by the equation that is just put in over here, and your Vmax equals to K2 into this. So you can substitute this and you can get that particular equation, Vmax into substrate concentration divided by Km plus substrate concentration. This is the Michaelis-Menten equation. So what happens at V naught equals to Vmax by 2. So at Vmax by 2, your Km is equal to substrate concentration. So finally, when you generate a plot like this, at different Km values are at different substrate concentration, at Km equals to substrate concentration. When Km equals to your substrate concentration, that is the time when Vmax or V naught equals to Vmax, that is the maximum velocity, divided by 2. So you can calculate it. So the Km is the substrate concentration, where the initial velocity equals to half of your maximum velocity. So the Km widely varies among the different enzymes. So the Km can be expressed as Km equals to K minus 1 by K1 plus K2 by K1, which equals to Ks plus K2 divided by K1. So as Ks decreases, the affinity of the substrate increases. The Km can be a measure for substrate affinity if K2 is less than that of K1 minus 1. That is the backward reaction. Okay, now we have a few plots that we need to study, and then we have the plots. We have the inhibition kinetics and the mixed kinetics, followed by this. Okay, we have a few more slides and then we will be solving the questions. So up till now, if you have any question, you can ask me. Okay, before we go into the plots, because now there is nothing much to understand. It is just generating the different kinds of plots, like Weaver-Berg plot and all. So but in the inhibition kinetics is also very important. Up till now, if you have any question, you can go ahead. Okay, so no questions. I guess it's clear. Good. So let's take just 2 minutes break and then we can start again with the plots. Just 2 minutes. right. So okay, now getting back once again. So the line we were so we have the double reciprocal plot. So this is nothing, just arranging the equation where you take the initial velocity at the denominator and then you frame the equation in such a manner where you can plot 1 by Vmax equals to 1 by V naught versus 1 by S, that is the 1 by substrate concentration on the x-axis and 1 by V naught on the y-axis, and then you get a straight line. So the thing that you have on the x-intercept is minus 1 by Km, and on the y-intercept you have is 1 by Vmax, and then you have a slope. So your slope equals to Km by Vmax. So this is your y-intercept, this is your x-intercept. So 1 by V naught intercept is equal to 1 by Vmax. The extrapolated x-intercept is equal to minus 1 by Km. So this is the Lineweaver-Burke plot. Slope equals to Km by Vmax. So for small errors, that is at low substrate concentration, leads to large errors in the 1 by V naught. So K catalysis equals to minus Vmax by total enzyme concentration. So Kcat is how many reactions an enzyme can catalyze per second, that is the turnover number. If we observe for the Michaelis-Menten kinetics, where K2 equals to your Kcat when the substrate concentration is very less than your Km, so very little complex is formed and your enzyme concentration is equal to the total enzyme concentration, because delta Es, that is the complex that is formed, is very less. So your entire enzyme is conserved. So V naught equals to K2 by Km into your total enzyme concentration into total substrate concentration, that is equal to Kcat by Km of enzyme concentration, enzyme and substrate concentration. So Kcat by Km is the measure of the catalytic efficiency. Now, a very important thing about the inhibition kinetics. So inhibitors are the ones that interfere with the substrate in different ways. So see, you have the succinate. Now some inhibitor comes and changes the shape, where you have one CH2 less and the succinate dehydrogenase cannot form the fumarate. So competitive inhibition, where the inhibitor competes with the substrate in the active site of the particular enzyme. So you have the enzyme, you have the substrate, this is the normal reaction that is happening. But apart from the substrate, you have an inhibitor. Now it's a competitive, either it binds to the substrate or it binds to the inhibitor. So if it binds to the inhibitor, the enzyme inhibitor complex is getting formed. And then when in the presence of the substrate, no reaction occurs. Okay, just give me 1 minute. Okay, so once again, so enzyme plus substrate, also in that particular medium you have an inhibitor. So what it does, your substrate, the binding of the substrate to the enzyme is interfered by the particular inhibitor. So the enzyme inhibitor complex is formed and the substrate cannot bind to the active site, so no reaction occurs. So the kinetics here stands that Ki equals to E into I, enzyme into inhibitor concentration plus the complex concentration. So there is an additional entity that is being introduced with the Km. So this alpha equals to 1 plus inhibitor concentration divided by the equilibrium constant of the inhibitor reaction. So what happens? So you see that This is where no inhibitor, then the higher the alpha value, your curve gets um, so with this your curve gets affected, right? So when you plot it in the Line-Weaver-Burks plot, you see that with the increasing inhibitor, the slope becomes a lot more steep. So you get a steeper curve, a steeper straight line compared to a no inhibitor. And then the slope also changes. It increases. The x-intercept is more shifted towards the positive side, and the 1 by Vmax remains the same. Now for an uncompetitive inhibitor, what happens is that the enzyme substrate complex is formed, and then comes the inhibitor that goes and binds to a different position other than the active site. So both the substrate and the inhibitor is bound, but in any ways the reaction won't be formed because it is also interfering the formation of the reactant. When it's a non-competitive inhibitor, both the x and the y intercept is hampered, but the slope remains same. And here another factor, that is alpha. You can see that alpha dash is equal to 1 plus this inhibitor concentration divided by your equilibrium constant. Finally, the mixed inhibition is there are two types of inhibitor. The inhibitor can either bind at the primary stage or after the complex is getting formed. In either case, the reaction will not be formed, but these are the two different stages. So it's a mixed inhibition. In that case, what happens, both the x and the y intercept is hampered, the slope also gets affected. So the two uh, different stages of inhibition by the two will be controlled by two variables, that is alpha and alpha dash, where for alpha dash is for I dash inhibitor and alpha is for the I inhibitor. So mixed inhibition is when the inhibitor binds to the enzyme at a location distinct from the substrate binding site. So the binding of the of the inhibitor will either alter the Km or the Vmax or both. So you see Ki equals to this and Ki dash is this. So where there is an additional substrate. So V naught equals to Vmax into substrate concentration divided by alpha Km plus alpha dash into substrate concentration, where your alpha dash is 1 plus inhibitor concentration divided by Ki dash, and your alpha equals to 1 plus inhibitor concentration divided by Ki. So the different types of inhibition. So if it's not no inhibition, Vmax remains Vmax and Km is Km. So app is apparent. Competitive, Vmax remains Vmax. Vmax is unchanged, whereas Km increases. Uncompetitive, Vmax decreases. Km also decreases. And for mixed inhibition, Vmax decreases, but Km may or may not be may be increased, may be decreased, depending on the alpha and the alpha dash values. Okay, so this is about the enzyme inhibition kinetics. Okay, I think we are good to proceed with the question and answers, right? Up till now, do you have any questions? Any questions on anything? You can ask me. Else, we can start with the question and answers. You do not have any questions? So I guess everything is clear. So you can answer all the questions, all the answers correctly. More or less all the answers correctly. right? Okay. Sounds fantastic. Great. Okay. So are you there? Yeah, good. So you can answer me the questions in the chat box, and then we can discuss. Okay. I think the questions won't be very tough. So let's start with the first one. For a given enzyme reaction, S plus E forms enzyme substrate complex, and you get an enzyme plus product. One should monitor the absorbance of the dash versus time. So what should monitor the absorbance of dash versus time? So what should be monitored? Is it the product, the substrate, tyrosine, tryptophan residues present in the enzyme, or tryptophan tyrosine residues present at the active site of the enzyme? Okay, you say it's substrate. But if you see from the kinetics, what does it tell? Which rate were you actually studying? It was dp by dt. So the rate of product formation that you want to study. So why should you observe the absorbance of a substrate that is actually minimizing? Because your substrate is finally getting consumed and your product is the one that is getting formed. So why would you want to check the rate of the forming or rate of the uh, consumption of a substrate rather than understanding the rate of formation of a product? So the correct answer over here is product versus rate, not substrate. Okay. Okay. Anyways, question 2. When the velocity of enzyme activity is plotted against substrate concentration, which of the following shape is obtained? It's a very good question, and you need to read the question once more. Velocity versus substrate. There can be different ways of framing the question. It's a very important question because there will be questions from the graph and the plot. So kindly read the question before jumping into an answer. So what will be the shape of that particular curve? Parabolic, hyperbolic, straight line with positive slope, straight line with a negative slope. Its velocity plotted against substrate concentration. Very good an answer. It's option B, correct. Hyperbolic. The next one is match the following. So match lyase, hydrolase, oxidoreductase and ligase. I just discussed about the functions of all these enzymes. Won't be very tough for you to answer. So let us see. You said it's D. So let's see what does do. Let's start from the last. Ligases. So catalyze reactions in two chemical groups are joined by the use of energy from ATP. Yes, so one. So 1, 2, 3. So option B is wrong. Next, let us see. So 2, 4 and 4. So 2. Act to add or remove hydrogen atoms. So that is the function of an, yes, oxidoreductase. So A and D may be right. Right. Then we have hydrolase. Hydrolase, oh, hydroxylase. So add water across a bond, hydrolyzing it. Very good. Fantastic answer. It's B, it's D. Sorry, it's D. So one lyase is add water, ammonia, carbon dioxide across the double bonds or remove these elements to produce double bonds. Hydroxylase is add water across a bond hydrolyzing it. Oxidoreductase carry out oxidation reactions by the action to add or remove hydrogen atoms. And finally, ligases catalyze reactions by joining the two groups with the help of ATP. Fantastic. The next one. For the reaction, substrate plus enzyme, then you get the product and enzyme. This is the substrate, enzyme, and product respectively. The quantities that can be measured experimentally are. So what are the quantities that you can measure experimentally? Substrate, product, total enzyme, bound substrate, bound product, bound enzyme, only substrate and product, only free product. Try to recollect the answer from the values that are probably constant or are either increasing or decreasing. So you need to measure the quantities experimentally. So what are the things that you can measure experimentally, or you already have idea about? Try to come up with an answer. This is not tough. This is easy. This is very easy. We all know it. So you have the substrate that you know the concentration. Let us assume a protein or an enzyme. Okay, let us consider a protein whose concentration you know, and at the end, if you check, you can check the value to be decreasing. So substrate is the one that you can measure experimentally, and you see that it has either consumed or not consumed. Whatever has happened has happened. The product is another thing that also you can measure. What about the enzyme? Yes, that is also you can measure, because the total enzyme is known to you, because you are only the one who is adding from outside. So the substrate is known, the product is known, and the total enzyme concentration is also known to you. So these are the three quantities that you can measure. Okay, right. Question 5. In which form the enzyme exists when the maximum velocity is observed? When the maximum velocity is observed, at in which form does the enzyme exist? Is it the enzyme form or the complex form or both the forms, or it's difficult to tell? Think and answer. So when the maximum velocity is reached, very good, means that the enzyme substrate in the enzyme substrate complex, because your enzyme is saturated with the complex and it cannot take up any further substrate, so that is where the maximum velocity is achieved. Which of the following is not a characteristic of enzyme? So tell me which of the following is not a characteristic of enzyme. Enzyme accelerates reaction by increasing the free energy of activation, binds to transition state of the reaction better than the substrate, exhibits saturation kinetics, and works at optimum pH and temperature. Yes, it's a very easy answer, because What is happening? Because by decreasing the free energy of activation, right? Yes. So A is not a characteristic of the enzyme. Question 7. In enzyme kinetics, the turnover number is defined as. So what is a turnover number? Read the options and give me an answer. So what is a turnover number? Maximum number of substrate that can be converted to product per molecule of that enzyme per second. So one molecule of enzyme in 1 second converting the number of substrate to product, that is the turnover number. Very good. So option A is the correct answer. Fantastic. Up till now it's going very good. So question 8. The diagram shows a lock and key model for an enzyme that has been affected by an inhibitor. What type of inhibition is happening to this enzyme? Okay, interesting. Wow, that was quick an answer, and that's the correct answer. So option B is correct. Competitive inhibition, because it's just blocking the way of the substrate by competing with that active site of an enzyme. Next one, which of the following equation is used in Lineweaver-Burke plot? Another very important and interesting question. That is why you need to keep all the equations in your mind and you need to learn them, practice them. There can be few questions from there. There can be numerical problems, NAT, anything can be there. Mostly for your this upcoming exam, you won't be having NAT, but for any other exams, competitive exam, this is a very very what you can tell. All the question setters want this type of questions to be there in the question paper. Yes, so D is the correct answer. Very good. 1 by V naught equals to Km by Vmax into substrate concentration plus 1 by Vmax. Where we are plotting 1 by V naught versus 1 by substrate concentration. Slope is Km by Vmax and your y-intercept is 1 by Vmax. Correct. So next one. In an enzyme catalyzed reaction, dash provides information on substrate binding and dash provides information on the reaction rate. So what provides information on the substrate binding and what provides information on the reaction rate? So you have Vmax, Km, Kd, 1 by Vmax, Kcat, 1 by Kd. There are a lot of variables. So find out the correct answer. Information on substrate binding and information on the reaction rate. Yes. So Km is the one from where you can get the information of the substrate binding and Kmax dominates the information on the rate of the particular reaction. Very good. Well done. So question 11. What it says, this is a Lineweaver-Burke plot for an enzyme that has been affected by an inhibitor. So the blue line is with inhibitor, the orange line is without inhibitor. So what form of inhibition is occurring to this enzyme based on the graph? Oh, you have already given an answer. Or that's for the other answer. Yeah. Yes, it's an uncompetitive inhibition. I just showed the graph. So it's a direct question. Good. Uncompetitive. A small organic molecule bound to an apoenzyme by a covalent bond. So what It is a small organic molecule bound to an apoenzyme by a covalent bond. What do you call it? Prosthetic group, complex enzyme, holoenzyme, coenzyme. Yes, it's a prosthetic group. Fantastic. Next one. Which of the following is just a minute. Just a minute. So which of the following statements is or are correct regarding RNase A? So you have RNase A. It catalyzes the cleavage of the phosphodiester bonds via the formation of trivalent phosphate transition state. In RNase A catalysis, histidine 12 and 119 have the ability to accept and donate electrons. The specific substrate for RNase is CCMP, cyclic CMP, and phenylthiourea is a standard inhibitor for RNase A. So if you see that RNase A catalyzes the cleavage of the phosphodiester bonds via formation of a not trivalent, it is pentavalent phosphate transition state. So it has a pentavalent phosphate transition state, not trivalent. So one is incorrect. And the histidine 12 and 119 have the ability to accept that. And the specific substrate for RNase A is cyclic CMP. So 2 and 3 are the correct options. So we have it in option D. So keep this thing in mind. This is wrong. This will be pentavalent. The next one. Which of the following is true for Michaelis-Menten kinetics? So which of the following is true for Michaelis-Menten kinetics? Something is given about Km, then about the covalent bonding and all. So which is true? The best thing about this type of questions is to find out which of the following are false. So you just have to find out the 3 false statements. No answer? Okay. So let us see what is Km. So Km is defined as the concentration of substrate at which enzyme is working at half of maximum velocity. Not at maximum velocity. It is at half of maximum velocity. It is also a measure of the affinity that the enzyme has for that particular substrate, and Michaelis-Menten kinetics assumes non-covalent binding between the enzyme and the substrate. Only thing that is true is it describes the single substrate enzymes. Only single substrate enzymes work for the Michaelis-Menten kinetics. That is only true. Other statements are incorrect. The last question, it's about a fruit. When a sliced apple is kept in open air, it becomes brownish due to what? Oxidation of phenolic compounds in apple by enzyme ortho-diphenol oxidase, reduction of phenolic compounds in apple by an enzyme ortho-diphenol oxidase, inhibition of enzyme ortho-diphenol oxidase, and inhibition of phenolic compounds in enzyme by enzyme ortho-diphenol oxidase. Yes, so it's an oxidase, so it will perform oxidation of phenolic compounds in apple by enzyme, that particular enzyme. Yes, so this is correct. Very good. So that is all from my side. If you have any questions, you can go ahead, maybe from this class or last classes. Okay, then, if you do not have any questions, we can end the class over here. Thank you for joining. We will again meet in the next week, same time, 6 p.m. for a new lecture. Okay, thank you. See you in the next class.